Good evening, students. Myself, Ankit Kumar Malik, PhD fellow, and then PMR fellow as well. So, today we'll be covering the integral doubt session for the topic chromosomal basis of inheritance, linkage, chromosomal disorders, classical genetics experiments, bacteria, and viruses, with clinicians' perspective 5. So, we'll start with the question 1. So, the question 1 asks that Ramesh was provided with specimens of gram negative and gram positive bacteria. So after performing the steps of gram staining, so he proceeded for observation under a microscope. So, but he saw both slight purple in color. So what could be the possible reasons for this? So the possible options for these questions are: so he forgot to add the crystal violet. Option B states that he forgot to fix the dye with the iodine. Option C states that he forgot to perform safranin staining. And the option D states that he forgot to heat the slides after adding crystal violet. Hmm. So for this, will hello. Okay, I think it's audible. So in this question, it seems like Ramesh encountered an interesting result during his gram staining experiment. So gram staining is a crucial technique in microbiology that helps differentiate bacteria into two major groups, that is gram positive and gram negative. So based on the characteristics of their cell walls. So the typical outcome of a successful gram staining procedure is that gram positive bacteria appears purple, while gram negative bacteria appears pink or red in color. So this color difference is due to the ability of the bacterial cell wall to retain the crystal violet dye used in the staining process. So if both the gram negative and gram positive bacteria specimens appeared purple under the microscope, so it suggests that there may have been an issue with the gram staining procedure. Let's explore a few possible reasons for this unexpected results. So the first uh, reason could be the first possible reasons is over decolorization. So decolorization is a critical step in gram staining where the crystal violet is removed from the gram negative bacteria. If the decolorization step is prolonged or too harsh, it can be result in both types of bacteria retaining the purple stain, leading to confusion. And another option could be old or improperly stored reagents. So the quality of the reagents used in gram staining is crucial. So if the reagents are old, contaminated, or not stored properly, so they may not perform as expected, so leading to inaccurate results. The third reason could be an inconsistent staining technique. So the gram staining technique requires a precision and consistency. If Ramesh did not follow the steps carefully, or if there were variations in the technique, it could affect the outcome. The next reason could be cell wall variations. So, in some cases, certain bacteria may have atypical cell wall structures that do not conform the typical gram positive or gram negative characteristics. So, this could lead to an unexpected staining. So, to address this issue, Ramesh should carefully review each step of the gram staining procedure, ensuring proper timing and technique. If the problems persist, he may need to consider the freshness and quality of the reagents or investigate whether the bacterial strains used have unusual cell walls characteristics. And troubleshooting the gram staining process will help him obtaining accurate results in distinguishing between gram positive and gram negative bacteria. So, according to these theories, so the possible answer could be the possible reason could this the option D, sorry, the option C. So, as the statement states that he after performing the steps of gram staining, he proceeded with for observation under microscope, but he saw both the slides purple in color. So the option C could be the correct reason that he saw both the slides purple in color. That is, he forgot to perform safranin staining. So let me explain you what safranin staining is. So safranin is used as a counter stain in gram staining. The counter stain is used after decolorizer in gram staining. So counter stain is used to detect gram-negative bacteria, as these bacteria do not retain the primary stain used in the gram staining called crystal violet. So the option C is the correct answer for the option for the question one. Moving forward to the question two. Questions two ask that suppose you are screening for an antibiotic which works for inhibiting peptidoglycan of cell wall. So you take two bacterial strains, that is A and B. You notice that A stains violet in gram staining and B stains red. So antibiotic will be effective against which of the following strains? Whether it is strain A, B, or both AB, or cannot be determined by gram staining experiment. So for this question, so in this context of the gram staining results and the mechanism of action of the antibiotic targeting and peptidoglycans in cell wall, let's analyze the situation. So here the gram staining is a technique that categorizes bacteria into two major groups. It is in the last question I've already told you that is gram positive and gram negative. So now here it is mentioned that the bacterial strain A stains violet in gram staining, which indicates that it is gram positive, whether strain B staining red is gram negative. So given that the antibiotic works by inhibiting peptidoglycan in the cell wall, so it implies that the antibiotic specifically targets and affects bacteria with a peptidoglycan layer. So gram-positive bacteria like strain a heavy thick peptidoglycan layer make them susceptible to the antibiotic. So therefore, the antibiotic will be effective against bacterial strain, a gram-positive strain that strains violet in the gram staining process. I'll show you the figure as well. So it may not be as effective against strain B. So the gram-negative strain that stains red as its thinner peptidoglycan layer is less affected by the antibiotic designed to inhibit the peptidoglycan synthesis. So coming back to the question, so according to the theories, so the option A could be the right one. So the antibiotic will be effective against strain A. The correct answer for the question 2 is the option A, that is strain A. So moving forward to the next question, that is which of the following conditions lead to the death of the mouse in the famous Friedrich Griffiths experiment? So there are some conditions. So the condition 1 is living S strain bacteria injected in mouse, living R cells injected in mouse. So there are two strains, that is R strain and R strains, S and R. So here S strains stands for smooth shiny colonies and R stands for rough colonies. So let me continue with the statements or conditions. Condition 1 is living S strains bacteria injected in the mouse. The condition 2 is living R cells injected in mouse. And the option C, the condition third is heat killed S cells injected in mouse. And the statement 4 for the question 3 is mixture of heat killed S cells and live R cells injected in the mouse. So there are some conditions. So they're asking in which condition the death of the mouse in the famous Friedrich Griffith experiment will be done. So we'll go through the famous Friedrich Griffith experiment first, then we'll come back to the question and answer this question. So Griffith experiment was a stepping stone for the discovery of genetic material. So Friedrich Griffith experiments were conducted with Streptococcus pneumonia. Streptococcus pneumonia. So during the experiment, Griffith cultured Streptococcus pneumonia bacteria, which showed two patterns of growth. One culture plate consisted of smooth, shiny colonies, while other consisted of rough colonies. So the difference was due to the presence of mucus coated in S strain bacteria, whereas the R strain bacteria lacked them. So the experiment in which Griffith has done the things. So in there, Griffith injected both S and R strain to mice. So the one which was infected with the S strains developed pneumonia and died, while that infected with the R strain stayed alive. Okay, so the moving further. So in the second stage, So Griffith heat killed the S strain bacteria and injected into mice, but the mice stayed alive. Then he mixed the heat killed S and live R strain. So this mixture was injected into mice and they died. So in addition, he found living S strain bacteria in dead mice. And the conclusion for this experiment is based on the observation. So Griffith concluded that R strain bacteria had been transformed by S strain bacteria. So the R strain inherited some transforming principle from the heat killed S strain bacteria, which made them virulent. And he assumed that the transforming principle is genetic material. So this was the Griffith experiment and its transforming principle. So coming back to the question, so according to this, the statement 1 and statement 4 are the statements which led to the death of the mouse in his experiment. That is condition 1 and condition 4. Stating living S strain bacteria injected in the mouse, and the statement 4 was mixture of heat killed S cells and live R cells injected in mouse. So the option A is the correct answer for the question 3. Moving forward to the next question, that is question 4. So here, the cannabar eyes are a recessive sex-linked trait in fruit flies. So what percentage of F1 males will have cannabar eyes if a female with cannabar eyes is crossed with a wild type male? So in this scenario, hmm, a sex-linked trait refers to a discernible feature of an organism that is impacted by the genes located on the chromosomes responsible for determining the organism's gender. So the X-linked recessive inheritance involves the transmission of a genetic trait or condition from parent to child due to mutation occurring in a gene situated on the X chromosome. So in males who possesses only one X chromosome, the occurrence of a mutation in the sole copy of the gene on the X chromosome, so which leads to the manifestation of the condition. So in this situation, every male individual will experience the same condition. According to this, the every male individual will experience the same condition. So according to this, the possible answer could be the 100%. That is option D. So the option D is the correct answer for the question 4. Moving forward to the next question, that is question 5. So in question 5, it states that Mahesh is suffering from a sex-linked recessive disorder, hemophilia. So he has a daughter who has normal phenotype, and she marries to a man who is of normal phenotype, but her mother-in-law is a carrier of hemophilia, and her father-in-law is suffering from the disease. What could be the probability of their daughter being hemophilic? So the probability are option A, that is 0, 0.25, 0.75, and 1. So we'll go through the scenario. So in this scenario, Mahesh is suffering from a sex-linked recessive disorder, that is hemophilia, which is carried on the X chromosome. Since Mahesh is affected, so he must have the genotype XHY, where XH represents the chromosome hmm, carrying the hemophilia allele, and the Y is the male sex chromosome. So Mahesh's daughter, who has a normal phenotype, which means she inherited a normal X chromosome from Mahesh. So let's represent the genotype as X and HX. So now the daughter marries a man with a normal phenotype, that is XY. However, her mother-in-law is a carrier and her father-in-law is suffering from hemophilia. So let's consider the possible genotypes of the daughter's offspring. So here the daughter's normal X chromosome combines with her husband's normal X chromosome, resulting in daughters with normal phenotypes and sons with normal phenotypes. So the daughter's normal X chromosome combines with her husband's Y chromosome, resulting in sons with normal phenotypes. So now let's consider the daughter's carrier mother-in-law. So the daughter's X chromosome combines with her mother-in-law's carrier's X chromosome, so resulting in daughters with a normal phenotype and sons with 0% chances of being carrier. So the daughter's X chromosome combines with her mother-in-law's hemophilic X chromosome, resulting in daughters with 0% chance of being carrier and sons with 100% chance of being hemophilic. So the probability of the daughter being hemophilic is 0%, assuming a random assortment of chromosomes during gamete formation and fertilization. So coming back to the question, after all these theories and calculation, so the here the probability of the daughter being hemophilic is 0%. So the correct answer for the question 5 is option A, that is 0%. So 0% is the probability of the daughter being hemophilic. So coming to the next question, that is question 6. So, so question 6 states that given below is the representation of a normal chromosome in which letters represent the genes and represents centromere of the chromosome. It's P, Q, R, S, T, U, V, W, X, Y, Z. So which of the following is the correct match of the given chromosome with their respective structural alterations? So here one statement is called alteration in chromosome structure. So we'll go through the theories first, then we'll come back to the question and we'll look into this. So the basic concept behind this question is alteration of chromosome structure. So alteration in chromosomal structures refer to changes or abnormalities that occurs in the arrangement, size, or number of genetic material within a chromosome. So these alterations can result from various genetic processes, including mutations, error during DNA replications, or exposure to certain environmental factors. So there are several types of alteration in chromosome structure, each with its own characteristics. So the first option is deletion removes a chromosomal segment. So here deletion involves the loss of a segment of a chromosome, and the consequences from the deletion step is the missing genetic material can lead to the loss of specific genes, potentially causing genetic disorder or developmental abnormalities. And the second step is duplication. Duplication repeats a segment. So here duplication occurs when a segment of a chromosome is copied, resulting in an extra copy of the genetic material. So the consequences of the duplications are that duplicated genes may lead to an overexpression of certain traits or contribute to the genetic variability. The next step in alteration of chromosomes is an inversion reverses a segment within a chromosome. So here inversion involves the reversal of the order of a segment within a chromosome. Understand? So the consequences with this step will lead to that while the gene consequence remain the same, the altered orientation may affect gene expression and disrupt normal genetic functions. And the last step for the alteration of chromosomal structures is a translocation moves a segment from one chromosome to another non-homolog one. So sometimes it's called translocation. So the translocation occurs when a segment of one chromosome breaks off and attaches to another non-homologous chromosome. So the consequences for the same step leads to result in the exchange of genetic material between chromosomes, potentially leading to gene disruption or altered gene regulations. So there are two more steps are there, but will not look into that steps that named as ring chromosome and isochromosomes. So we'll not look into that two steps that are not that important. So these four steps, so these structural alterations can have significant consequences on an individual's health and development depending on the extent and nature of the chromosomal changes. So they may lead to genetic disorders, birth defects, or increased susceptibility to certain diseases. So understanding these alterations is crucial in the field of genetics, and health researchers and healthcare professionals comprehended the underlying causes of various genetic conditions. So moving back to the question, which what the question states. So according to the theories and the basic concepts, so the Correct answer for this is the option only one, that is deletion step. In deletion, I've told you that deletions involves the loss of a segment of a chromosome. Here, the missing genetic material can lead to the loss of a specific genes, potentially causing genetic disorder or development abnormalities. So the right option for the question 6 is the option A. There is only one. The deletion step would be there. Moving to the next question, that is question 7. So the question 7 states that let's assume a bacterial cell which is initially growing and replicating in 14N, that is light band, containing media is transferred to 15N, that is heavy band containing media. So what would be the composition of DNA of bacterium after two generation in 15N medium? So in this scenario, the scenario you are describing here involves the use of isotopic labeling of trace DNA replications in a bacterial cells. Let me break down the process for you. Wait. Hmm. Here the initially the bacterial cell is growing and replicating its DNA in a medium containing the lighter isotope, nitrogen 14. This means that the nitrogen atoms incorporated into the DNA molecule are predominantly of the 14N isotope. Here the spelling of E. coli is wrong, just ignore it. That's E. coli, C-O-L-I. They've mistaken dot coil. No worries. Just ignore that mistake. Now the bacterial cell is transferred to a medium containing the heavier isotope, that is nitrogen 15N. During DNA replication, the new strands will incorporate the available nitrogen from the medium, which is now predominantly composed of the heavier 15N isotope. So after one round of DNA replication, that is from first generation, the resulting DNA molecules will be a mix of the original DNA strands labeled with 14N and the newly synthesized DNA strands labeled with 15N. So this hybrid DNA composition is referred as a hybrid, or you can say intermediate density. So now when the bacterial cell undergoes a second round of DNA replication in the 15N containing medium, the following scenarios can occur. That is conservative replication and semi-conservative replication. Hmm. That is one is a conservative and another one is semi-conservative replication. So in conservative replication, if DNA replication were conservative, the original 14 and labeled DNA strands would remain together and the newly synthesized 15 and labeled strand would form a separate set. In this case, you would have two distinct populations of DNA, one with the only 14N and another with only 15N. 14N and 15N. And the next scenario is semi-conservative replication. So in this replication, if DNA replication follows the semi-conservative model, each DNA double helix would consist of one original 14 N labeled strand and one newly synthesized 15 N labeled strand. Okay, so this results into two DNA molecules, each composed of one strand from the original DNA and one newly synthesized strand labeled 15 N DNA, 15 N only. So concluding this statement, coming back to the question. So according to the theories and principle and basic concept for the same, so we'll look into the question. So so the composition, that is 15 and 50, the 50% and 15, 14 and 50%, the option B would be the composition of DNA of bacterium after two generation in 15 and medium. So the option B is the correct answer for the question 7. So moving to the next question, that is question 8. So it states that a syndrome is caused when a group of symptoms occur together and characterize a particular abnormality or condition. Turner syndrome is a genetic disorder caused by abnormal cell division. So what chromosomal abnormality is the cause of the disease, whether it is trisomy 21, trisomy 18, trisomy 13, or trisomy of X chromosome. So we'll look into the disorders first. What trisomy is, what Turner syndrome is. So Turner syndrome. Hmm. let me explain first Turner syndrome. So Turner syndrome is a genetic disorder that results from a chromosomal abnormality affecting the sex chromosomes. So individuals with Turner syndrome typically have one normal XX chromosome instead of the usual pair, that is XX. So the chromosomal abnormality associated with Turner syndrome is the partial or complete absence of one of the X chromosome. So now I'll explain you what trisomy syndrome is. So a normal human usually have 23 pairs of chromosomes with the two sex chromosomes, and that determines and or 44 which determines sex, and 44 autosomal chromosomes which are responsible for growth and functions. So trisomy is a genetic condition caused by an alteration in the number of chromosomes where the affected person has three copies of one of the chromosomes instead of two. So the affected person possesses 47 chromosomes instead of 46. So trisomy 13, there is one condition that is also called Patau syndrome. So trisomy 13 or Patau syndrome is the result of having 13th chromosome in a person's DNA three times as opposed to twice. So trisomy is a rare genetic disease. Trisomy 13 affects a child's face, brain. Wait, let me show you. Just one, a Patau syndrome. So, trisomy 13 affects a child's face, brain, and heart development, in addition to causing physical growth issues throughout the child's body. So, trisomy 13 can have deadly symptoms and raises the risk of miscarriage or death before the child turns 1 year old. So, moving to the possible options, the options D, trisomy of X chromosome. Let me tell you what trisomy of X chromosome. So it is referred to a genetic condition in which an individual is missing one of the X chromosome in the typical pair. In humans, females typically have two chromosomes and males have one X and one Y chromosome. So trisomy of X chromosome occurs when an individual, regardless a normal human usually has 23 pairs of chromosomes with two sex chromosomes that determine sex and 44 autosomal chromosomes which are responsible for the growth and functions. So according to these theories, so the option D is the correct answer for the question 8, sorry. So trisomy of X chromosome is the cause of the disease, which is a syndrome caused when a group of symptoms occur together and characterize a particular abnormality or condition, Turner syndrome. Okay, so moving to the next question, that is question 9. So the question 9 states that Rashmi was initially growing bacteria in 15N, that is heavy media, 15N, that is heavy band, and 15N media, and then she moved them to the 14N, that is light band media. So let us assume that DNA replication follows conservative model. I've told in the last question, last to last question. So what conservative model is, what the unconservative. So let us assume that DNA replication follows a conservative model. Then which of the following bands would appear after density gradient centrifugation after two generations? So the possible options for the question 9 are, so one band of light DNA and one band of DNA with medium density. And option B is one band of heavy DNA and one band of DNA with medium density. The option C, one band of light DNA and one band of DNA with heavy density. And the last option for the same questions are a DNA band of DNA, a single band of DNA with medium density. So let me put some light on the conservative replication model first. So in this scenario, wait, let me to the question. So in this scenario, Rashmi initially grew bacteria in a medium containing the heavy isotope, that is 15N, and then shifted them to a medium with the lighter isotope nitrogen 14. So if DNA replication follows the conservative model, we need to consider the expected bending pattern after two generations of bacterial growth in the 14N medium. So let me put some light on the what conservation replication model is. So this conservative replication model suggests that after replication, one of the resulting DNA molecule is composed entirely of the original strands, while the other is composed entirely of newly synthesized strands. So therefore, the two DNA molecules formed during replication would have different densities due to the isotopic difference. So in its purple, it's original DNA strands, and in blue, it's newly synthesized DNA strands. So after two generation, in the 14N medium, we would expect to see two distinct bands in the density gradient centrifugation. So in heavier band, so what? Let me tell you what heavier band shows and what lighter band shows. What 15 and what 14N is. So the 15N, that is heavier band. So this band represents the original DNA strands that were present before the shift of the 14N medium. So these strands can contain the heavy isotope nitrogen 15 and have a higher density. So on the other hand side, lighter band, that is 14N. So this band represents the newly synthesized DNA strands that incorporated the lighter isotope, that is nitrogen 14, that is 14N, during replication. So these strands have a lower density. So coming back to the question, so concluding this question, after two generations in 14N medium, the density gradient centrifugation would reveal two bands, one heavier band containing the original 15N containing DNA, and on the other hand side, one lighter band containing the newly synthesized 14N containing DNA. So considering the options, available options, so the right answer for the question 9 is the question C. There is one band of light DNA and one band of DNA with heavy density. So the right option for the question 9 is the option C. So moving to the next question, that is question 10th. So here brown wings in Drosophila, that is represented by capital B, is dominant to white wings, that is represented by small b, and the red eyes color, that is represented by capital R, is dominant to white eyes color, represented by small r. So what fraction of the progeny of the cross of this combination will have brown wings and white eyes? So let us see. So here some combinations are given here, so we'll just calculate this. So according to this calculation, so brown wings and white eyes genotypes are capital B, double capital B, and double R, capital B small b, and double small r. So according to this calculation, the option A is the right answer for the question 10, that is 50%, that is 1 by 2.
డైరీ మెక్కిందా మరి మంచిగా ఫోన్ తెచ్చుకోలేదని వచ్చేసి నేను కొంచెం
Shishira, your mic is, can you just mute your mic? Okay. So the option A is the right answer for the question 10. That is the half fraction of the progeny of the cross of this combination will have the brown wings and white eyes. So this is for the day. So thanks for attending the session, students. You can now leave the session one by one. Thanks for your time. Shishira, you can leave the meeting. Sure. The session will be uploaded in the YouTube. So you guys can leave the meeting now.